This method can help in the pre-selection of promising clones of Pichia Pastoris strains expressing recombinant proteins under de-repressed conditions by simple shake flask cultivations prior to upscaling. The main advantage of this technique is that high-level methanol-free protein expression can be achieved using Pichia Pastoris, which is supported by the exact online monitoring of important cultivation parameters. Generally, individuals new to this method may struggle because the time point for starting the glycerol feed has to be adapted to the growth characteristics of the specific production strain.
Before setting up the main culture, seed a single fresh colony of the expression strain of interest into 5 milliliters of yeast peptone dextrose broth in a 50 milliliter conical tube. Leave the lid slightly open to allow proper aeration. Then place the culture in a shaker at 28 degrees Celsius, 80% humidity, and 100 to 130 RPM overnight. The next morning, start the software for connecting the devices via Bluetooth. Next, press the silver button on the front of the biomass measuring device to turn on the Bluetooth connection to the device, and click Find Devices in the computer monitoring software. Drag and drop the found devices to the squares in the measurement tray and click Connect. When a successful connection is made, a control screen will appear. To set up an experiment, click Start Measurement. Name the experiment and check if all parameters required for the monitoring are enabled. Set the interval to 3 minutes and the average measurement points to 11. Enter the names for the samples and skip the